Hello everyone, welcome back to the session on rodent behavioral models and setups. So the last session we covered various behavioral models, examples of behavioral models, and we started off with the commonly used behavioral model in neural engineering, that is stroke model. So in this stroke model we have gone through the manifestations and clinical features and what are the features that one should look for to replicate in the rodent. So we have introduced Whishaw test into the topic, and then we will see how the Whishaw test helps in replicating the manifestation of the stroke, and why is it so important to have an objective observation in order to make sure that your research objectives are met. All right. So this is where we stopped last time. This is the skill reaching rating scale. So as I said earlier, the Whishaw test will have around 10 steps where the rodent's task of pellet retrieval through a slot in the in the box. So the rat has to use its forelimb, pass the forelimb through that slit that is provided, and then grab the grab the pellet and then put it in its mouth. So the entire behavior of this single pellet retrieval has been broken down into 10 different steps. All right. So it's very important that you video record it, and how the cameras are arranged around the behavioral setup box has been explained earlier. So this is the importance. In order to capture these individual 10 different steps of that single task is of paramount importance. So for example, you may think the stroke behavior will cause hemiparesis or hemiplegia, so it is really easy. But then if at all your stroke surgery is suboptimal, and if the rat is able to still reach the foot pellet, and then the task itself becomes a success, then you would wonder, is there any real difference before the stroke and after the stroke? And that is when this kind of objective rating would come into picture. All right. So every single steps should be recorded, and compare it with your pre-stroke behavioral training videos, and see if there is any subtle differences among these 10 different steps. So it's basically a more objective way of looking at the behavioral test rather than just estimating the behavior just by visual inspection. And if there were no these 10 different steps, you would say that okay, rat is able to grab the pellet and then feed itself, then you would consider that behavior as normal, and you will be wasting a very precious subject. So obviously you need to observe the movement that are occurring at the level of digits, and see if the digit moves to the midline, and then is it really semi flexed, and then is the elbow is reaching the midline. And that is why the angle of your camera matters so much. You need to observe all these subtle differences in the posture of the limb as well as the body of the rodent. And what exactly is Arpeggio? Is this pronation really complete, or is it really impaired? So that point to point comparison is pretty much important to make sure that there is some manifestation of the stroke surgery, and rat does have some weakness in performing the task. All right. So, and obviously if there is an intervention planned for the rat that is having some disability through this objective rating, then obviously you can easily compare the outcome or the improvement if you are subjecting the rat to any sort of management, like interventional management in terms of be it a medication or be it physiotherapy exercises that you can try and exercise the muscle which is weak and then see the improvement, or rather if you are trying to bring in some translational neural engineering into picture where you can really compare with your post intervention recordings. So that's where the entire these test parameters matters so much. All right, so let's see how these 10 different steps does look like, and this is just an example. It may not be just Whishaw test, it may be even a lever press task, or even the head is fixed and then there is a rotating disc where the rat is grabbing the food pellet. So any task for that matter, you need to have some sort of objective rating or objective observation for the behavior that you are going to use it to validate your any experimental setup. So that is the whole point that I am trying to go into so much of details as to why and how that you would observe the behavioral test in detail. All right. So now let us look at these 10 different steps, how it would appear and why is it so important. And these two pictures you can clearly make out that this is the front camera and this is the camera from below. Alright. So there are some views where the side camera is going to be very beneficial, and the camera shot from below, I mean this is the snapshot from below which is very important, and you need to have some sort of a reference midline. So you are trying to see that whether the digit has been brought to the midline. It is a preparatory step for the full grasp of the movement that is going to happen. So you need to see that this is the midline and that is the midline of the rodent body. So is it really approaching the midline or not is something you need to really understand. So we need to look at the movement that is occurring at the level of shoulder, elbow and the wrist of the rodent, and that is a preparatory step happening where the forepaw swings to the midline because the slit is here, and that is the food pellet. If you can make out that it has to pass through this slit, before that happens the rodent prepares itself and brings the digits to the midline. And if you see the movement of the long event rats, you would really wonder, are there any such different steps, because all you see is rat comes and within a fraction of a second it grabs the pellet and puts it into its mouth, and that is where your digital recording is going to help, and so that every frame need to be analyzed. So the camera that you are going to really use should have high frames per second and high shutter speed. So that is very important, because once you record the video you need to analyze frame by frame and capture this particular step, that is digits to midline, and various other steps that is going to come into future discussion. So that is where it matters so much, where the rat brings it into the midline and then only the next step happens, that is the semi flexion of the digits. So that is again it is making its hand smaller in girth so that it passes through that slit. So that is another minor movement, that very precise movement that is going to happen in its digits, that it is semi flexed, alright, as opposed to the extension that is happening to the other digits. If you compare, it is semi flexed. This is the best comparison that you can make. This is the fully extended digit. You can see how this is semi flexed. Every single digit has been semi flexed, so that it becomes globular in shape and can easily pass through the slit. That is the whole idea. So that is the next step that is happening after it brings it to the midline. And the third step is the entire elbow moves to the midline. Alright. Initially what we saw, digit comes to the midline and then the elbow also comes to the midline, and that is a wonderful example how important that our posture of the body changes with sequence of movement. All right. So, this is where we usually call in humans ergonomics. In order to make the movement efficient, you will arrange your body and your footstep automatically, so that your hand makes a finer movement which is close, which is small and pretty precise. So, here the precise movement is that it is going to pass the hand through the slit. That is a very precise movement, and then it has to grab the pellet and then retrieve it back. So, in order to perform that precise task, the rodent has to arrange its forepaw and its body, balance it, and then do all these movements so that the next step happens. So, that is the whole idea. So, this is the another step where the elbow itself comes to the midline. First the digit comes to the midline, whereas the elbow will be a bit lateral, and as the movement progresses, the elbow as well comes to the midline. Next is the advance. So, in this step, the rat try to pass the forepaw into the slit. At the same time, the snout goes up. That is the advance that is happening. It advances its limb by putting the nostrils upwards. It shifts its nostrils upwards so that it makes space for the forepaw to come. At the same time, it is balancing with the other forepaw on the ground. So, that is the beauty, and that is where the precise movement of the distal forelimb comes into picture, and that is why this is going to give us a lot more neural signals and lot more information about the distal forepaw as opposed to any other tasks that are available. So, once the initial advance is made, next is, if you remember, the third step was digit semiflex. Now it is going to extend it. All right. So, that is the digit in full extension. So, these are very minor differences in its digits and elbow and body, which it is difficult to observe and then make a direct observation and direct it is very difficult to make an observation and record it just by visual inspection. That is why your camera and the analysis of the video post capture is going to help us immensely. Alright. So, there are automated ways of doing it, and there are automated analysis which we will be discussing it in next few slides, but then you need to understand these movements in order to analyze it with any other software, because these are all anatomical changes that are occurring in the forepaw of the rodent in order to grasp the foot pellet. So, it extends the digit, and then before grasping it, the hand goes on top of the foot pellet in a movement called pronation. All right. Pronation is the hand goes palm down on the surface. That is the pronation movement, and the opposite of that movement is supination. Okay. So, you need to be familiar with the word prone and supine. So, the prone is the natural position of the rodent body itself, that is prone. When the ventral surface, that is when the belly comes on the top, that becomes a supine position for the rodent body itself. Whereas for the hand, that is the dorsum, palm is ventral, that is dorsum of the hand. So, that when the dorsum of the hand is on top, that is known as Arpeggio. Alright. So, very important to observe this particular movement, because as I said, if your stroke surgery is incomplete, or the infarct is incomplete, or the rat is recovering very well, then there may be only subtle differences in these kind of finer movements that you are going to observe. So, it may be that the arpeggio is incomplete, maybe that it is not really fully extended, limb is still in semiflex position, maybe that each and every movement is taking more time than what it was observed before inducing the stroke. So, arpeggio is the pronation movement where it keeps its extended digits on the top of the foot pellet. The natural consequence after arpeggio is grasp movement, which you can easily make out that it grabs the food pellet with complete flexion of all the digits, alright. That is very important, and that is where this particular rat species, this particular test, everything sort of resemble closely with what we do. Though we have individuated digit movements where only the thumb and index can move, where the opposition can happen, those are all really special human movements. But then in rodent, this is the closest that one can see. So the seventh rating parameter is the grasp movement. Once it is grasped, as I said, there is this supination movement going to happen. Alright. So once the food pellet is grabbed, the next natural step is to retrieve it into the box and then feed it itself, feed the pellet itself. So obviously need to reposition its forelimb. The first movement what has happened was the Arpeggio, where the rat keeps all the digits on top, then it gradually rotated back, which is called supination movement, alright. So supination is just the opposite of the pronation, as I explained earlier. But then the supination does not happen in one flip like that. Obviously the rat's hand is going to get stuck there if the full width comes into the area of the slit. So what it does is there is a semi supination that happens, or semi prone position, where it is tried to withdraw the forepaw, which is called supination 1. Then is the supination 2. As soon as it withdraws the food pellet into the setup box and then it has to feed itself, that is when the complete supination happens, alright. So that is supination 2, where you can see that the hand is completely supinated position. So that is supination 2. Supination 1 is outside the box, supination 2 is inside the box feeding it itself. So then obviously the release, which is the last step, where the food pellet is released into its mouth before it starts chewing and swallowing. So those are the 10 steps, 10 individual steps that you can observe, break it and then analyze it and compare it with each and every component that you have captured before the stroke surgery or after your interventional strategy comes in. So that is how you can rate the entire behavior. For that matter, any behavioral test you can break it down into 10 different steps or 15 different steps, as long as there is a consistency in it, because you are going to do around 30 to 40 trials of these pellet retrieval every day for almost 7 days. Alright, so there are good amount of data, video data, that you can analyze and see how consistent it is. So that is something you need to do before you do the stroke surgery. Though there are 10 steps in Whishaw test, if suppose you do not get long Evans rat and if you are using some other species, or for that matter maybe the rat itself, but you are not seeing these steps consistently, but if you at least try and make or at least 7 to 8 steps consistently, which is capable of reproducing itself every time you repeat the task, then those are the parameters that you need to choose. So any test that you might take or any species that you take, there has to be some consistency throughout your experimental plan, starting from the training period, interventional period, or the post intervention analysis time. So entire period this step has to be repeated and then compared, alright. So let us look at the automated apparatus. So as I said, it is bit tedious to have the single pellet retrieval task, and every time you need to do many things manually. For example, if you do not really have help and you are the only person who is going to conduct the experiment, where you need to handle the camera recording, you need to record on your note as well, and then look at the food pellet retrieval that is happening simultaneously by the rat. So there are multiple sequences that are occurring in tandem. So then how would you help yourself? One way is to have a automated apparatus where there are infrared detectors attached to this behavioral setup box, and then there are infrared LEDs that are there next to the food pellet tray. So every time some movement is detected, there is a pellet dispenser here which delivers the pellet into the tray, and then the rat will grab it and then move backward, and whenever the movement is detected again, another pellet is detected. As soon as the rat tries to grab the pellet, the camera recording will be switched on by the IR LED which are placed outside the box right on top of the food trays. So there is a camera recording going instantaneously as soon as the rat becomes active in grabbing the pellet, and then there is an automated dispenser which dispenses the pellet as soon as the movement of the rat is detected. So there are all the important sequences that are supposed to happen in the Whishaw test can be reproduced here, and all that you have to do is to record the movement that are happening and then make sure that nothing goes wrong, though the apparatus is automated. So just your supervision is required, and the rest of the things will happen on its own. So that is the pellet dispenser, IR detector, camera that has been mounted on the box. So there are many such automated apparatus that are available. You can look around for these kind of setups as well. Now let us look at another task which is a very advanced way of looking at it. Advanced in the sense, here is the task where the head is fixed. I hope you all remember the cranial window that we discussed during the surgery, that surgery sessions, wherein the cranial window is created by doing a craniotomy and then fixing a metal plate on top of it, so that you can use 2-photon microscopy on top of it. Alright. So then the rat's head is fixed with the stereotactic frame there, and then you have a cranial window to look at the neurons in action. Alright. You are trying to study the neuronal activity while the rat performs various tasks. So that is the beauty of this head fixed cue multi-step prehension task. So prehension is basically grabbing the food pellet. That is the task that it's similar to the single pellet retrieval task or Whishaw test, except that here we have the rat's head fixed to the cranial window, and the microscope is used to visualize the neuronal activity. Basically you will see a fluorescent activity going up, and you can even study through the layers, and various neurons will light up as and when the rat performance activity. And that is the, you all can look up the principles of 2-photon microscopy, where there is a calcium spike that happens every time the rat makes some activity, and that induces fluorescence in the neuronal population, and that is captured through your microscope. Alright. So then each and every movement can be captured using a video camera, and then you have an infrared camera here where this can give us a beautiful automated analysis of the kinematics that are involved in the movement of the forepaw. Alright. So this is how it is. Every time a cue appearance, an audio cue, that is where the speaker is there, every time a cue happens, the disc rotates, and the pit with the food pellet comes closer to the rat for it to grab. So it rests the forepaw on a horizontal metal bar until the food pellet reaches the rodent's, closer to the rodent's mouth. head is tethered here or fixed to the cranial window, that is the cranial window. So, this is the side profile. We can see the hand moves and makes a trajectory and then takes the foot pellet, just similar to the Whishaw test. And what are these colored blobs? These are what is known as kinematic analysis. So, this is done by a software called Deep Lab Cut, alright. And how this is done we will see. So, basically there are multiple trials that are going on, and in every single trial more or less the rat's forepaw takes the same path for it to reach the foot pellet and then feed it, feed itself. So, that is what is analyzed here, and this is one step higher than the Whishaw test rating scale, where there is an objective analysis and then there is some amount of artificial intelligence involved here, because you are trying to create an algorithm wherein the software picks up the movements that are happening it automatically and then gives you the trajectory after averaging through multiple trials that the rat grabs the foot pellet. Alright, so that is how the task is performed in this particular behavioral apparatus. So, the advance that has happened with this particular task is that it is markerless tracking of the digit. So, when you look at the complexity of the behavioral apparatus, you have visual analysis, which is a basic kind of analysis where you capture the video and you go through those 10 different steps and make sure you visually analyze and visually compare, are there any differences between the pre-stroke and post-stroke, alright. The second step is that you put a marker on the rat's forepaw. For example, either you color it and then follow the colored hue, movement of the colored hue and then analyze it. So, there some marker is involved, or you can even tie IR markers called gluons, infrared markers which are reflective markers on the forepaw of the rodent, and then use your infrared camera to analyze the movement. That is the second level of sophistication that one can see. But then obviously it is very difficult to maintain such markers. If it is a staining that you are trying to look, that might fade away, or if the infrared markers that you are going to use, that might come off the rodent when it moves around. So, markerless tracking is the next level of sophistication, where you are using AI algorithm to train the software, where you have a data to train and then say these are the markers that I am going to use by the anatomy, by the position of the limb, and once that training is done, the software automatically analyzes the trajectory as it is shown in this picture here, alright. So, these are the 3 different levels of sophistication. So, if one would ask which one should we really prefer, I would obviously say it is the third which is most sophisticated, not just because there are no markers which can introduce various errors, and it is also easier to analyze and more objective as compared to the visual analysis of your videos, alright. So, obviously if there is a way, if there is a chance to use this markerless tracking of digit and then use this software called Deep Lab Cut, then that is something very reliable, because it makes so much sense when you try to analyze the neural signals that are coming out. So, these neural signals are in the time scale of milliseconds, and your video steps that you are seeing probably are in millisecond, and these are microseconds. So, when there are milli and microseconds time scale involved, then it is all the more important that you have a higher frame per second video capture, a higher shutter rate in the camera. So then obviously your analysis should also match the efficiency of the camera. So that is where all these automated softwares and AI algorithm is going to make a huge difference when it comes to the objective analysis, and you can sort of easily synchronize the digit movement along with the neural signal. So in which step what sort of neural spike are you getting, and that would help immensely for decoding the neural signals that you have captured. So if there is some decoding is involved in the entire neural data capture, then you need to make sure that your video and the neural signals all are synchronized. That is very, very important. If it is not, then your video is trying to look at a different time scale, and the RAD's forepaw is moving in a different time scale, then obviously you are going to miss out on a lot of neural signals. So that is the whole idea, where you need to sort of capture all the movements, capture all the neural signals, and make sure they are synchronized. Alright. So that is very, very important. So in next session we will deal with markerless techniques. So thank you all for your kind attention.